In our final part of our exploration of the innate immune system, we're going to discuss the effector functions that are mediated by cells of the innate immune system once they get into tissues. We'll discuss a little bit about local and systemic inflammation and the broad effects that they induce, and we'll discuss what happens when there are certain aspects of the innate immune system that fail. So now we're going to focus on cells of the innate immune system, such as phagocytes like macrophages and neutrophils and natural killer cells. We won't discuss things like eosinophils or basophils or mast cells at this point. However, be aware that they will use very similar mechanisms to get into tissues, but some of their effector functions may differ. So this is a macrophage. This macrophage is recognizing pathogens that have been able to get into the tissue through an insult to this epithelial barrier. I like to think of these macrophages as our tissue alarm system. They recognize the pathogen and they make inflammatory cytokines such as interleukin 1 or tumor necrosis factor. This leads to the migration of neutrophils and monocytes into this inflamed tissue and migration of these neutrophils and monocytes along a chemokine gradient to the site of infection. These neutrophils I like to think of as our first responders. They are the first to migrate into an inflamed tissue, and they are the most abundant in inflamed tissues. Monocytes are also present in these inflamed tissues, except that once they migrate across the endothelial barrier into the tissue, they differentiate into macrophages. So monocytes can be found in circulation, but not in tissues. Macrophages are found in tissues, but not in circulation. This slide illustrates some of the key effector functions of innate immune cells that we are going to discuss going forward. We're going to discuss phagocytosis, which is mediated by phagocytes such as macrophages, neutrophils, and eosinophils. Degranulation, which is largely mediated by granulocytes such as neutrophils, mast cells, basophils, and eosinophils. The production of extracellular DNA traps by neutrophils, and direct cellular toxicity mediated by natural killer cells. So first, phagocytosis. Phagocytes are able to bind to pathogens via phagocytic receptors. These phagocytic receptors can bind things such as bacterial sugars and lipids. However, some of these phagocytic receptors can bind opsonins, and the two primary opsonins that exist are C3b and IgG. And these opsonins dramatically enhance the ability of these phagocytes to bind to pathogens and ingest them. Once these phagocytes ingest these pathogens, they take them up into this structure called a phagolysosome, where they can then destroy the pathogen. It's important to note that some bacteria are very resistant to phagocytosis because they have polysaccharide capsules. In this case, opsonins are critically important for control of these pathogens. And I've listed a few of those pathogens here. You may not have discussed these pathogens in any great detail in your education thus far, but when you come across them, you will learn more about their polysaccharide capsule and be aware that these pathogens require opsonins for their clearance. Once these pathogens are taken up into phagolysosomes, they can be destroyed. Phagolysosomes are formed when a phagosome fuses with another endosomal vesicle called a lysosome. The lysosome is highly acidified, and so this leads to an acidified phagolysosome, which can activate a number of enzymes, proteases, lysozyme, lipases, nucleases, etc., that can degrade the pathogen. There's a couple of other important things also that happen within the phagolysosome. One of those is oxidative burst. This is the production of reactive oxygen species and requires the function of this molecule called NADPH oxidase. A second function of the phagolysosome is nitrositive burst, and this is the production of a free radical called nitric oxide. These are highly damaging to pathogens and are critical for control of pathogens and destruction of pathogens within phagolysosomes. Many granulocytes, once they get into inflamed tissue, release their granules. These granules are full of things like lysozymes, molecules such as myeloperoxidase or reactive oxygen species that have highly toxic effects, inflammatory mediators such as leukotrienes and histamines. And much of what you see in an inflamed tissue, the redness and the swelling, comes about because of these degranulation events. Neutrophils are also expert at releasing their DNA. And as neutrophils die, they release DNA and then into what are called extracellular traps. These extracellular traps are rich in antimicrobial products that can trap and kill bacteria. I would note that the toxic effects of oxidative burst and degranulation means that activated neutrophils typically die within 24 hours of migrating into an inflamed tissue. So they are consumed rather rapidly. And in fact, the pus that you find in some bacterial infections is mostly dead neutrophils. The last function that we'll talk about is direct cytotoxicity mediated by natural killer cells. Now, natural killer cells are primed to kill any target they come across. However, a healthy cell expresses a number of inhibitory ligands that prevent killing by natural killer cells. But an infected cell that becomes stressed and expresses a number of molecules associated with that stress, called activating ligands, can lead to the activation of NK cells and the ability of NK cells to now release granules and poke hole in the target cell, allowing for delivery of those granules that induce apoptosis of their target. Therefore, natural killer cells are particularly important in eliminating virally infected cells. So the local inflammatory response is basically what we've been describing. This is induced by cytokines and complement components that lead to the infiltration of cells of the innate immune system into tissues where they can mediate elimination of the pathogen. However, under certain circumstances, when inflammation is strong and or prolonged, these inflammatory cytokines can go the other way. Instead of drawing cells in, they are produced in such high quantities that they get into the bloodstream and are distributed systemically. When that happens, there's a number of things that happen related to systemic inflammation. So what are the features of local and systemic inflammation? Well, the local effects of inflammation we've discussed. This is the activation of local immune cells, endothelial cell activation, leukocyte recruitment, chemotaxis of leukocytes to the site of pathogens, and macrophages would be considered here the early warning system or the alarm system. Neutrophils are the first responders. But what about when inflammation goes systemic? When inflammation is systemic, this is because of the prolonged production of inflammatory cytokines and their systemic distribution in the blood. One of the things that can happen is this leads to release of neutrophils from the bone marrow so that there's increased neutrophils in circulation. So if you have somebody that has systemic inflammation and you do a CBC, they should have increased neutrophils. They may also have increased neutrophils because systemic inflammation leads to increased hematopoiesis, or the production of new leukocytes in the bone marrow. If prolonged, the systemic inflammation can also lead to the entry of immature neutrophils or bands into circulation. This is a sign of particularly prolonged systemic inflammation. Systemic inflammation can lead to fever, it can lead to rashes, it can lead to something called the acute phase response in the liver, which we'll talk about in detail in a separate session. And it can lead in extreme cases to sepsis and septic shock. Sometimes acute systemic inflammation can be chronic, or even within a tissue, inflammation can be chronic. And there are a number of diseases associated with chronic inflammation. And in fact, regardless of the specialty that you end up in medicine, it is very likely that at some level you will have to deal with the pathologic effects of chronic inflammation. This includes things like inflammatory bowel disease, cardiovascular disease, obesity, and even immune dysfunction during aging. Now, what happens when the immune system doesn't work? There are a number of failures of the immune system that can teach us about the importance of each of these steps that we have discussed of the innate immune response. So, for example, deficiencies in the alternative and classical complement cascades can lead to susceptibility to infection with encapsulated bacteria, such as Neisseria meningitidis. If you'll recall, we mentioned that encapsulated bacteria can really only be phagocytosed when they are coated with opsonins such as C3b. When there are deficiencies in complement inhibitors, this can lead to overactive complement activation, and this can include diseases such as paroxysmal nocturnal hemoglobinuria, which is the sporadic lysis of red blood cells due to overactivation of complement. Deficiencies in pattern recognition, such as deficiencies in toll-like receptor signaling, can lead to susceptibility to infection with a number of things such as fever-inducing or pyogenic bacteria. Defective in the oxidative burst within phagolysosomes can lead to a disease called chronic granulomatous disease. And this is due to mutations in the NADPH oxidase that lead to susceptibility to a wide variety of bacterial and fungal infections. And defects in lymphocyte trafficking can also lead to susceptibility to infection. The canonical example of this is leukocyte adhesion deficiency, which is a deficiency of LFA1 and leads to a failure of leukocytes to traffic into infected tissues. And again, this leads to susceptibility to many bacterial infections. As I mentioned, in a separate lecture, we will discuss in greater detail the effects of cytokines on both local and systemic inflammatory responses.